Assalamu alaikum everyone, this is Hani, and today in this video I will talk about differences between eukaryotic and prokaryotic translation. So let's start it. The first difference is the polymerase used in the translation. In prokaryotes only one polymerase is used, but in the eukaryotes several polymerases are used. The second one is the ribosomes. In the prokaryotes 70s with 30 and 50s subunits of ribosomes are used, but in the eukaryotes the 80s and 40 and 60s of subunit of ribosomes are used. The third one is the transcription and translation process. In the prokaryotes, they can overlap because keep in mind that in prokaryotes there is no nucleus, no nuclear envelope. So the translation and the transcription is occur in the cytoplasm, and they are continuous process, so they can overlap. But in eukaryotes they don't overlap because the transcription is occurring in the nucleus and the translation is occurring in that cytoplasm. So the next is starting amino acid. In prokaryotes, the N-formyl methionine is the starting amino acid, and in the eukaryote, the methionine, only methionine is the starting amino acids. The next is the initiation factor. In prokaryotes, IF1, IF2 and IF3 are only the prokaryotic initiation factor, but in the eukaryote, EIF2, EIF3 and the subcategories of EIF4 are the eukaryotic elongation factor. Here the E means the eukaryotic, I means initiation, F means the factors. To describe the eukaryotic initiation or elongation factor, we write a small e before the factor. So the second is the elongation factor. EFTU is and EFTS and EFG are the prokaryotic elongation factor, while EF1 and EF2 are only eukaryotic elongation factors. So the next is the termination factor, which is also known as the release factor. So we describe or write as the RF. R mean release and F mean the factor. So prokaryotic release factors are RF1, RF2 and RF3. But in the eukaryotic release factor there is only one release factor, which is RF. And the next is the initiation and termination sites. So as in prokaryotes transcription and translation is continuous process, so translation and transcription sites in prokaryotes are several, but in eukaryotes the only single site is the initiation and the termination site. So the last one is the synthesis of transfer RNA. Is in the prokaryotes, the synthesis of transfer RNA is from DNA or RNA, but in eukaryotes it is from only RNA because of the presence or occurring of the transcription and translation in different places. So this is all about a difference between eukaryotic and prokaryotic translation. Thank you for watching. I hope you will learn about difference between translation in prokaryotes and eukaryotes from this video. Thank you for watching.